Understanding of molecular biology is driven by the central dogma, which demonstrates how DNA, which can self-replicate, is transcribed into RNA that is then translated into functional protein. Ordinary task that is similar to this process is ordering fast food. Your verbal order represents the original message within DNA. This order is transcribed into the visual instructions for the restaurant. This intermediate step would be the messenger RNA. That provides the template for the order, which represents our protein. How do the visual instructions become the food? The answer is the employee, which performs a job similar to tRNA, also known as transfer RNA. This molecule will transfer the genetic message into functional proteins, just like the worker will transfer the order from the screen to the final prepared meal. Behind tRNA was first postulated by Francis Crick. Essentially, his idea was that there be a molecule that would facilitate the linking between amino acids and nucleic acid due to the lack of a direct interaction. This adapter molecule, which he believed to be RNA, would be able to read the incoming codon from the mRNA within the reading frame on one end, while carrying the proper amino acid on the other, which would add to the growing chain. To our fast food example, each codon, which represents one item within the full order, the employee tRNA would read one sequence codon at a time, make the proper item, then add it to the ongoing order, just as each amino acid is added to a growing chain. What we finish is with a completed fast food order or a polypeptide chain. A closer look at tRNA in its unique clover shape. At its 3' end, an ester bond will form between the molecule and an amino acid. At one of the 3 loops is the anticodon sequence. So let's look at the steps of translation. Ribosomes are the home of translation. mRNA strands enter the ribosome, then tRNA will bind to it. The codon within the reading frame will bond to an anticodon on the tRNA. When bound, the amino acid comes into the picture, which code for the peptides that are added to a chain that will be folded into proteins. However, errors in the original DNA can produce alterations in the amino acids. These can come from internal or external sources, such as incorrectly nucleotide placement or UV damage respectively. This can lead to a plethora of mutations during translation, as shown by the table on the right. The most severe case would be an error in the production of a start or stop codon. If a start codon is mutated, then translation will not occur at all, and no protein will form. A mutated stop codon will lead to either premature or delayed termination. In both cases, the likely result is cell death, as vital proteins essential for proper cell function would be lacking. However, the process of wobble can prevent the alteration of amino acids. This phenomenon is illustrated by the figure on the left, as there are multiple nucleotides within the third position that can code for the same amino acid. For example, GCA, GCC, GCG, and GCA all code for alanine. It did not matter what nucleotide was in the third position.